Hola, buenos días, buenas tardes a todos. Vamos a dar inicio ahora con el seminario institucional y esta vez, pues, el gusto de que nos presente Michael algunos de sus avances en su grupo de investigación. Entonces, bueno, tienes la palabra, Michael. Gracias.
Gracias, Jesus. And thank you for the invitation. So today, I'd like to talk about a bit of what we're doing using alfalfa as a model plant to study interactions with a couple of different types of bacteria. Okay. Yeah, okay. So first of all, to just kind of define the term polyamines, these are small hydrocarbon compounds that have varied numbers of amino groups, which, because of their positive charge at physiological pH, can have interactions with different types of macromolecules such as amino acids, proteins, or phospholipids, and they've been implicated or shown to affect a lot of different physiological processes in organisms. Much of the work has been done in eukaryotes, but also they are very important in plants and prokaryotes. So speaking of plants, polyamines have been shown to affect a lot of processes in plants, either exogenously applied or in genetically manipulated plants that overexpress polyamines, affecting various physiological processes that result generally in higher yields or more tolerance to biotic or abiotic stresses. Likewise, in bacteria, in particular, this table talking about rhizobia and the possible functions of polyamines in rhizobia, a lot of things have been described as being dependent or influenced by polyamines. But surprisingly, most of these studies did not involve genetically defined mutants of rhizobia, and there are more correlations between polyamine levels and different phenotypes. In the field of study of plant pathogens, there is more evidence for the involvement of polyamines produced by the pathogen or by the plant host in modulating the pathogenic interactions. This is Pseudomonas syringae pathovar tomato. And the finding of the paper was that if you knock out the ability to synthesize spermidine in the bacterium, then it becomes more resistant to challenge with peroxide. So there's sort of an inverse relationship. It's not really understood why this should be so, but that was the main finding of that paper. Here, looking at effects of polyamines, specifically in Cynorhizobium meliloti, we see that they generally affect phenotypes such as biofilm formation, exopolysaccharides, or motility, really the kind of thing that polyamines are known to affect in many different types of bacteria. So today I'll be talking about some of the work that we have done characterizing various mutants either in signaling systems or in the production of specific polyamines. This is a scheme of polyamine biosynthesis, kind of a conglomeration of different pathways that exist in different organisms. The pathways with the red arrows are more or less the canonical system that occurs in eukaryotes, for example, and the ones in blue are what is known as alternative systems. And these exist in many plants and some bacteria. For example, Cynorhizobium has most of the blue pathways, and also some of the red pathways that deal with arginine metabolism and ornithine metabolism, which we have studied in previous years. Most of the focus that I will have today, as far as different polyamines, will be on this putrescine, which is sort of a precursor polyamine for spermidine and also for homospermidine. So those are three of the major polyamines I'll be talking about. And this just illustrates a symbiotic phenotype of a mutant that we call ODC2 that is deficient in polyamine production, and its phenotype with alfalfa is to form very sick-looking small plants. You can complement this phenotype by also feeding the mutant inoculated plants with putrescine, which is the main polyamine that they lack, and putrescine is the precursor for the other polyamines that are lacking. So you are able to complement chemically this phenotype. So now to move on to the polyamine signaling part, we know that you can, that both endogenous and exogenous polyamines affect phenotypes in Cynorhizobium meliloti. So one question is, if polyamines that are present in the environment, such as root exudates, also modulate phenotypes in Cynorhizobium. And so we have seen that alfalfa root exudates contain various different types of polyamines whose production is influenced by inoculation with Cynorhizobium. But most of our results have been somewhat inconsistent, partly due to the problem of obtaining root exudates in some sort of fairly natural manner. Another question is there polyamine exchange between roots and Cynorhizobium. We know that Cynorhizobium can excrete low levels of polyamines into culture media. As far as we have determined, this is not influenced by adding root exudates or inducers such as luteolin. And so the question I want to address here is, how would polyamine signals to S. meliloti be perceived and transduced? And for that, we took a page from studies that were initiated in Vibrio cholerae, where they found a polyamine sensing system that also affected phenotypes in Vibrio. And that consisted of a polyamine sensor that is called NSPS and a enzymatic activity encoded by MBAA that works to either synthesize or degrade the second messenger cyclic dye GMP. And so what they found in Vibrio basically is that the presence of homospermidine promotes the formation of biofilms, and spermine promotes a more planktonic lifestyle. And so in Vibrio, in the situation, in the absence of exogenous polyamines, you have sort of a moderate production of cyclic di-GMP and a moderate formation of biofilms. If norspermidine is ligated to NSPS, which has homology to a putrescine substrate binding protein, you get high production of C-di-GMP by activating the activity of this motif responsible for C-di-GMP synthesis, high levels of C-di-GMP that promote biofilm formation, whereas with spermidine, you have low production of C-di-GMP due to more activity of this degradative function, and you have lower biofilm formation. I think putrescine is sort of kind of a null effect for this system. So anyway, just to restate the effects of cyclic di-GMP, normally it promotes biofilm formation, whereas high levels inhibit motility. So you have an inverse relationship normally between biofilm formation and motility. So this I just copied from basically the Vibrio system. We have a NSPS homolog in Cynorhizobium that is annotated as a spermidine putrescine substrate binding protein. It is predicted to be in the periplasm, and its neighboring gene is a phosphodiesterase that is a homolog of the MBMA of Vibrio. So it seems from evidence that I'll show, perhaps the main activity of this complex here is to degrade cyclic di-GMP, which would influence production of biofilm, exopolysaccharide, and nodule numbers. And so just to look at some details, we looked for genomes that had neighboring NSPS and MBMA homologs. In red are Cynorhizobium and Cepher species, and the rest of the species that contain these neighboring genes are Pseudomonas and, as you would expect, Vibrio. So it's fairly well confined to Cynorhizobium strains among the Rhizobia. In Cynorhizobium, the genes occur together. As I said, the enzymatic part of the complex has domains for both synthesis and degradation of cyclic di-GMP. David Zamorano ran some experiments using the cloned MBMA and NSPS genes in a system in E. coli with a C-di-GMP biosensor to look at the effects of expressing the Cynorhizobium MBMA or NSPS on cyclic di-GMP levels. And from this data, when expressing only the MBMA, there is a decrease in cyclic di-GMP. When both NSPS and MBMA are expressed together, there's a further decrease. So it would seem that under the conditions of these assays, MBMA appears to function as a degrader of cyclic di-GMP. Now, another thing we considered is that quorum sensing in Cynorhizobium and other bacteria has a big influence on processes. that are influenced by cyclic di-GMP. So, for example, I'll talk about two Cynorhizobium strains, strain 1021 and strain RM8530. Strain 1021 has a natural insertion in a gene encoding a transcriptional regulator that's called EXPR. This is an important regulator of quorum sensing, and so effectively strain 1021 is deficient in quorum sensing, whereas strain RM8530 has a copy of EXPR without the insertion, and it is functional. So the production of acetylhomoserine lactones in Cynorhizobium is dependent on an activator, and these AHL signals can bind to the product of the EXPR gene, where they will influence various processes such as exopolysaccharides and motility, and also the production of quorum sensing signals. So you have a interlocking system of influences here. And for this reason, we looked at both the function of NSPS in 1021 and RM8530 to compare it in the absence and presence of quorum sensing capabilities. And so we did see that as far as the transcriptional expression of NSPS, the sensor part of the system, it is much higher in the quorum sensing proficient RM8530, with the exception of assays done in the presence of exogenous spermine, where you have quite a large differential. In strain 1021, it is generally lower, but it is differentially affected by, for example, putrescine in this case. So then Victor Chavez did a lot of work characterizing the response of wild type and corresponding NSPS mutants of either 1021 or RM8530 under conditions where they were given exogenous polyamines or no polyamines, in terms of their ability to form biofilms or EPS, which is what I'll talk about here. Autoaggregation is another phenotype we looked at. In most cases, the response of the wild type and mutant strains are very similar to different polyamines. Only in some cases do we see a fairly marked difference, indicating that the NSPS system is responsible for some of these phenotypes. And rather than go through the graphs, this is just a summary of biofilm and EPS phenotypes in the different strains. So in strain 1021, quorum sensing deficient, we found that the presence of putrescine diaminopropane or spermine caused NSPS dependent differences, which were decreased biofilm, decreased exopolysaccharide production, and the inference here is that it probably increased the degradation of CDI-GMP in this situation. In the RM8530 strain, which is quorum sensing proficient, norspermidine was able to increase biofilm production and decrease EPS production, whereas interaction with diaminopropane increased both biofilm and EPS production. And so these changes probably result from changes in differences in the activity of the MBMA unit as far as synthesizing or degrading cyclic di-GMP. looked at, well, Victor looked at motility differences if you inoculate a plate of agar medium with a low concentration of agar in the center, the cells will swim to form a zone of growth. And depending on the diameter of the zone, we can say that some strains are better at swimming than others. Swimming is a movement that's dependent on flagella, either in liquid or in low agar concentrations. So this is just an illustration of what the swimming assays look like. There's another type of motility, swarming, which is surface motility, which occurs on semi-solid surfaces. So you can use agar with a higher concentration or medium with a higher concentration of agar to look at the ability of the inoculated strain to move in this sort of irregular type pattern. So with these data, it was found, again, a comparison of the different behavior of wild type versus mutant in swarming or swimming assays. So in this case, in the ability to swarm, we found that in 8530, the QS proficient strain or 1021, norspermidine had the effect of increasing swarming motility. As far as the swimming assays, we didn't detect any change that was different between the NSPS mutants and the wild type in either strain. And in contrast to Vibrio cholerae, where you have certain polyamines that have a reciprocal effect on biofilm and motility, we didn't find any polyamines that had that type of reciprocal effect in Cynorhizobium. Victor also did some thermal shift assays with purified NSPS protein. The idea here is that you mix it with a fluorescent, mix the protein with a fluorescent compound, and as the temperature increases and the denaturalization of the protein occurs, the fluorescent molecule is more available to give its fluorescent signal. And so you have a curve sort of like this. And in the presence of a ligand such as polyamines, these will tend to stabilize the structure of the protein. So for these assays, the profile for the control without polyamines was here. It has a fairly, it's fairly prone to degradation at a fairly low temperature, whereas in the presence of spermidine or cadaverine, you have a slight shift in stability and an even larger shift with other polyamines. And within this group of polyamines that appear to have a high binding affinity, you will find the ones that were also able to affect the phenotypes for EPS production and biofilm. So it appears that NSPS is a fairly broad, has a fairly broad substrate specificity for binding. And finally, what is the symbiotic phenotype of the NSPS mutants for the two wild type strains of S. meliloti? Well, probably not a great deal of difference in most of the parameters. Probably the largest difference that might be statistically significant might be in nodule dry weights between 1021 and 1021 and SPS mutant, but they are certainly not symbiotically defective under the conditions tested. Okay, on to transport systems, which is a newer project. We know that S. meliloti grown in cultures will take up and accumulate any polyamine that you add to the culture, which implies that there's either general or specific transport systems for different polyamines. We would think that polyamines need to be transported into the cell in order to cause physiological effects. This is not the case with the NSPS system because this interaction occurs in the periplasm. And so, but for other things like chemical complementation of mutants that are deficient in polyamine synthesis with exogenous polyamines, they have to transport them across the inner membrane, which requires transporters. And so one question was why S. meliloti has many polyamine transporters, as we'll see. And so we asked the question regarding polyamines in Cynorhizobium, that polyamine transporters would allow for intracellular levels to be modulated, as well as sending and receiving polyamines as signals. And in addition, we think that certain transporters in Cynorhizobium might function in acid stress resistance. So these were two of the questions, functions in signaling or in acid stress resistance, that we started to look at. So by analysis of the genome sequence, we found 11 different polyamine transport systems, including antiporters for different polyamines and cognate basic amino acids, a lot of ABC transporters, and a lot of transporters that were preferential for spermidine, at least according to the annotations in the database. A lot of these are designated by the POT designation, which stands for polyamine transport. The ones that are designated ABCD generally transport preferentially spermidine, but also putrescine, whereas POT, FGHI are normally specific for putrescine. And so we have in this collection only one right here, which is supposedly specific for putrescine transport only. And so we've mostly based on the work of Alejandra Arteaga, mutagenized a couple of our transporters. The POTF is the putrescine specific transporter, which biochemical assays show appears to be the main transporter for putrescine, but over a time course of hours, you can see the induction of other systems which slowly allow the transport of putrescine. So it's not the sole putrescine transporter. The other that was mutated, a putrescine agmatine antiporter, which would exchange these two polyamines. And the third one that was mutated was on the symbiotic plasmid B, a spermidine putrescine ABC transporter. As far as the symbiotic phenotypes, we checked those. There doesn't seem to be any great alteration as far as the ability to have a symbiosis with alfalfa. What we really need is mutants in multiple transport systems to try to knock out the this effect of system redundancy that Cynorhizobium has. As far as acid resistance in bacteria in general, they often use polyamines as part of an acid resistance mechanism. And these involve different antiporters for an amino acid and their decarboxylation. Here we are. basic amino acid and their decarboxylation product, which is a polyamine. And these reactions in the cytoplasm will consume protons and so raise the pH of the medium under acid stress conditions. And you also have some of these systems that are linked to metabolic pathways such as the arginine deaminase pathway, which produces ammonia, which alkalinizes the cytoplasm, also has the benefit of producing a bit of energy in the form of ATP. And so these, some of these systems correspond to ones that are found in Caenorhizobium, and in particular, the symbiotic plasmid encodes of the 11 transporters, more than half of them. And some of these transporters, such as a couple of antiporters, are clustered in a region along with decarboxylases and also genes encoding the arginine deaminase system. So we think these probably are functioning in acid stress resistance. We've done some work on these as far as a possible lysine cadaverine antiporter could be this gene, 0682, which we have shown does have some increase in its transcriptional expression under mildly acidic conditions. Close by, there's a gene 0684, which is a possible lysine cadaverine antiporter. Another possible system is composed of ornithine decarboxylase 1, which is an activity we have studied previously, and there is a putrescine ornithine antiporter, which is 0678, which is encoded close by. We know that inactivating ODC1 doesn't change the acid resistance phenotype of Caenorhizobium. We also know that the expression of ODC1 is probably similar to 0684 because they appear to occur in an operon. 0680 doesn't have a promoter sequence. And so the expression is probably equal to 0684, where we saw no change in expression at more acidic pHs. So the system work really remains to be done on the involvement of polyamines in acid resistance. As far as transporter gene expression, we have some data for 7 of the 11 systems. It seems that induction by polyamines for most of the systems indicates a wide substrate specificity if induction is equivalent to the substrates transported, which for transporters, many times it is. We do see an increased expression for some of the transporters in a mutant background where there are low endogenous levels of polyamines. So perhaps these transporters are increased in their expression because it's a benefit to the cell to transport more polyamines inside. So for pending work, in place of all the Gus fusion assays that we've been using, we'd like to do some quantitative reverse transcriptase PTR to look at PCR to look at selected transporters and the effect of exogenous polyamines on their transport characteristics. We could use the thermal shift assays that we used with NSPS to look at the substrate binding capability of the substrate binding proteins of different transporters, and also generate mutants in multiple systems to look at their phenotypes when they are actually deficient in polyamine transport. So now to look at another topic. All rhizobia make the polyamine homospermidine, whereas Caenorhizobium metalloti is essentially unique in being able to make spermidine. And also it does make homospermidine. So we asked the question of whether in S. meliloti, both of these polyamines that are present had redundant functions, or do either of them have specific functions in Cynorhizobium? So this just shows that rhizobia, apart from Cynorhizobium, do not produce spermidine, whereas all rhizobia produce homospermidine, and Cynorhizobium has both compounds. We inactivated in Cynorhizobium this pathway to make homospermidine by inactivating the HSS gene, and the ability to make spermidine by inactivating this gene that's part of the spermidine synthesis pathway to make single mutants, and also double mutants where both of these were inactivated. Then we looked at growth phenotype of the double mutant that is inactivated in both genes, so it cannot make either homospermidine or spermidine. We see that it has a growth defect, which introducing the empty vector here has no ability to complement the defect. A plasmid that is expressing the ability to synthesize homospermidine improves growth significantly, whereas expressing the gene allowing the synthesis of spermidine basically gives a wild type growth profile. So in this case, spermidine seems to be much more effective at complementing the growth defects of the double mutant. This is just to illustrate that putting these plasmids that contain either the homospermidine synthesis or spermidine synthesis genes do in fact result in an overproduction of the corresponding polyamine. So it's an effective system of genetic complementation, whereas in the case of the double mutant containing just the empty vector, it is devoid of both of these polyamines. So for another phenotype to compare, we looked at swimming assays of the different mutants. These are the single homospermidine mutants, single spermidine mutants, or the double mutant that cannot synthesize either, and compared their ability to swim without added polyamines or in the presence of exogenous polyamines, or in the presence or absence of the corresponding genes that can complement the defect in polyamines. Basically, what we found here is that only the double mutant that synthesizes neither is defective in swimming ability. It has a value of 15 versus 25 for the wild type. So then we went on to compare the ability of clone genes to correct the phenotype. Basically, with the single mutants, we're not really seeing a swimming phenotype defect. So we'll just zero in on the double mutant that has a clear phenotypic defect in swimming. And we found basically, for example, production of homospermidine from a plasmid is able to correct the swimming phenotype largely. It's a comparable value to that. And also production of spermidine is able to correct it. And essentially the same kind of data for complementation with different polyamines in the assay medium. So in this case, either polyamine seems to be effective for motility. some preliminary assays on swarming, just showing that the double mutant is able to swarm less than the wild type. Also, in terms of biofilm formation, it forms less biofilm. So these are other phenotypes we could look at with complementation experiments for these two polyamines. That's something to check. Yeah, that would be a good experiment. As far as symbiotic phenotypes, the double mutant is quite deficient in a lot of parameters, except for it forms somewhat more nodules. It makes smaller plants. This data indicated that it's probably an equivalent level of nitrogen fixation. The mutant plants or plants inoculated with the mutant are in the front. You can see they're smaller and less robust than the wild type, or of course the uninoculated mutants. I'll mention here that the HSS single mutant is not symbiotically defective, and experiments to determine the phenotype of the CanStack single mutant are in progress. Finally, Klebsiella vericula is a very common species of Klebsiella found in many environments. And the idea here was to use the type strain as a model organism to look at plant growth promoting traits. Now, many bacteria are able to do activities such as nitrogen fixation or produce phytohormones, which help plants grow or resist stress. And there's been a whole lot of studies on these systems with different bacteria. So we're looking at a collection of strains which was provided by Ulysses Garza, who has studied Klebsiella in detail with respect to multi-drug resistance, plasmids, and things like that. We have a collection of mostly clinical isolates. The plant-associated isolates here are in red, including the type strain that we want to use as a model. Most of these don't have a high level of antibiotic resistance. They were taken from various plant environments, and practically all of these have been sequenced. In fact, Klebsiella vericula was fairly recently used or developed as a commercial inoculant for corn, where they did some genome editing to allow it to fix nitrogen in the presence of fixed nitrogen in the soil, and in field trials saw increases in corn yield. So it's, even though many Klebsiella species are pathogens, Klebsiella vericula is present in many clinical samples from infected patients, but probably not as a cause of the infection. Further work done by Ulysses Garza's laboratory that shows that Klebsiella vericula has potential applications in biotechnology, of course, in plant growth promotions, and a lot of other things. So what we did rapidly is to start looking at plant growth promoting traits. Indole acetic acid production is an important component of many growth promoting bacteria. And so we inactivated the indole pyruvate carboxylase gene to knock out this pathway towards indole acetic acid. I should mention also, of the collection of 28 strains, we've screened most of them for indole acetic acid production, and the vast majority produce a similar level, whether they're clinical isolates or associated with plants. So it seems to be very common. the ability to produce IAA. So recently, work done with Alma Reyes to produce a mutant, and actually 3 mutants were isolated that basically do not produce indole acetic acid in cultures. The assays are done on the supernatant, so the indole acetic acid is being exported to the environment. So with this mutant, we can try to continue looking at what effect the absence of indole acetic acid production has on plant growth promotion. So far, it has not been very easy to demonstrate plant growth promotion by Klebsiella verricula in alfalfa, at least as a model. We've had rather variable results with that. Near-term goal is to look at IAA deficient mutants, perhaps versus IAA overproducing strains. Also, Klebsiella could be inoculated on alfalfa along with Cynorhizobium to look at a combined effect, and future studies perhaps with other growth promoting traits to try to look at what their participation in growth promotion is. So as a sort of summary, we, as far as signaling, we think NSPS MBMA does constitute a system of signal perception in vitro, at least, whether it's functional in nature and responds to, say, polyamines in root exudates is another question. Polyamine transporters, there's seems to be a large substrate overlap, and whether any of them have conditional expression conditions or fulfill certain functions is not yet known. As far as the 2 polyamines, spermidine, homospermidine, being interchangeable in S. melliloti, they seem to be largely interchangeable. So this question of why both are present is subject to more study. And as far as Klebsiella, we need a good plant model. Hopefully alfalfa will work out. And we'd like to define other PGPR traits because there's actually been surprisingly little work done on gene-directed mutants of plant growth promoting rhizobacteria. And a question here, whether distinctions between the clinical and plant-associated isolates is really very wide, except for the multi-drug resistance. So I think a lot of the clinical isolates might also have growth promoting abilities. Okay, just to finish up, thanks to, well, many people, former lab people who did studies beginning with the looking at polyamine precursors through current lab people. Hopefully I haven't forgotten to acknowledge the work that has been done by people and collaborators, both here in CCG and outside. So thank you.
Is there any question?
Thank you for the talk. I'm very interested in polyamines, so this was like a perfect overview for me. You mentioned it briefly in one slide, but could you elaborate on the effect of NSPS mutant on the symbiosis?
Excuse me, on
the symbiotic phenotype when you mutate NSPS?
Oh, when you mutate it?
Yeah.
It only has, you mean on general phenotypes?
Yeah, I mean, if you look at the plant or if you counted nodules.
No big effect on symbiotic phenotype, no effect on growth, no effect on polyamine profile of production of. So only the response to polyamines for phenotypes that are affected by polyamines, such as biofilm motility. That's what we've looked at.
And I have a second question. So because you have 11 transporters of polyamines, and you mentioned that you wanna keep doing mutants. Given that they have so much overlap, how are you thinking on doing the mutant, or what's your plan on how to overcome the big overlap?
Yeah, to make the multiple, technically, I guess it's possible to make multiple
like-
mutants
You
and
think you
perhaps
can-
select
Sorry.
groups that seem to be the redundant ones, like the spermidine putrescine transporters. There's like 5 of those. And technically, I guess one could use transduction, or Rafael Diaz has worked on a CRISPR system that might be useful, or by traditional methods of gene inactivation and replacement. So it's something we have to work harder on.
Thank you.
Si no, voy a preguntar. Gracias. When you do your thermal shift assay with NSPS, you seem to show that for some polyamines, they're binding. Was this done at a certain concentration, or did you vary the concentrations? Do you have an idea, is this high affinity binding or not?
I don't know how quantitative the binding assays were. Victor, do you remember what concentration of polyamines were? Or Victor Chavez.
Yo creo que la concentración quizás ronda los
It-
0.1 milimolar, quizás.
In the binding assays that Victor did, the other Victor.
Debe de ser 100 milimolar, me parece. Tenemos varias concentraciones, pero probamos hasta 100 milimolar y se ve bien el ensayo.
Pero no probaron las diferentes concentraciones para determinar cuál es una constante.
Probamos hasta 100 milimolar, pero probamos desde 0.1, 0.1, 0.5, 10, hasta 100.
¿Cuáles son los constantes de asociación?
No tengo el cálculo, solo falta.
You said you had mild acid conditions. What pH is that?
Oh, we were looking at either 6 or 5.5 as compared to 6.8, which is our normal.
And if there's a scheme and different polyamines, and in some case you said you wrote sim almost permitting. That's what I think I said.
That's,
What does that mean?
I believe it's symmetric because it has an equal number of let's find. Has amino-butyl groups that come out, so it's symmetrical, and they often call it sim. Is that what you're referring to?
I don't remember that had
Sim homosper, I'm just kind of abbreviating it, whereas spermidine has aminobutyl and an aminopropyl, so it's not symmetrical. And homo just means one methylene unit more than spermidine.
What temperature do you test the swimming or swam with kilograms?
Temperature,
Yes.
30 or 29 or 30 degrees.
Human cells grow at 37 degrees. Klebsiella does not fix nitrogen. 37, so maybe it is interesting to test different temperatures because they must live in humans. They must
Oh, I'm sorry,
grow
you,
at 37, and we have tested conditions of nitrogen fixation for the wild type and other plant-associated bacteria, and they can fix nitrogen at 37.
Were you asking about the plants?
No,
With
it's
Klebsiella?
about the spermidine, eggs, swarming or swarming at
yeah.
different temperatures.
Those were at all at 30 degrees. We haven't tried different.
Different temperatures. Yeah.
Mike, congrats for your talk. It was very interesting and your contribution, and congrats also for your contribution the last years about this producing subject in rhizobium. And I had a question, or maybe it's more a comment. When we was working a long time ago, you remember this project when we look into elements or compounds that restore the growth or minimal medium on rhizobium in minimal medium episode called. We test different compounds and use polyamines, putrescine, spermidine, etc. And when we grow the rhizobium in minimal medium, in liquid medium with polyamine and spermidine, we detect a opposite effect. I mean, these compounds affect the growth in minimal medium of rhizobium, decreasing the yield, maybe 5% or 30% of the final yield regarding to the minimal, the rhizobium growing only in minimal medium without polyamines. Do you have idea about what happened in this situation? Why the polyamines affect the growth of the minimal medium in this condition?
Specifically,
Mhm.
no. We know that for cynorhizobium with at least for 0.1 millimole polyamines that we generally use in our
Yeah.
experiment, growth curves are essentially unchanged. There's no effect. But cynorhizobium can use spermidine, putrescine, or cadaverine as nitrogen sources.
Okay.
So it can use them as nutrients also.
Regarding your mutant, the use of the mutant or the optin, the carboxylase, the phenotype on symbiosis, when do you restore spermidine, the sperm in we use spermidine to try to restore the phenotype of the type of the symbiotic. But do you know why this phenotype is not restored totally with putrescine?
No.
Because in comparison, this mutant with spermidine restore the nitrogen fixation apparently is restored with the supplementation, but it's not like to the wild type. Do you not have idea why the supplementation with putrescine is not restored totally?
In the, okay. Most of, that was Victor Becerra's experiment. I believe the supplemental putrescine seemed to be mostly transported to the leaves, rather than having maybe a direct complementation of the mutant that was being inoculated. Maybe it was affecting more plant physiology than.
Thank you.
There are no more questions. Acknowledge the mic.
You never check what is going on with the bacteria? You never check what